My name is Rishika Podarala, and this is my research on the isolation and identification of effective polyethylene degrading bacterial species from Galleria mellonella. The accumulation of plastic is becoming detrimental to the environment and human health, but previously researched microorganisms such as Ideonella sacchensis help facilitate the process of biodegradation. Polyethylene is a man-made material, which makes it an uncommon target for microorganisms to degrade in the environment. The aim of my project is to determine which bacterial species extracted from waxworms is most effective at degrading polyethylene. I predicted that bacteria grown on BHI and EMB agar will have higher percentages of biodegradation. The purpose of my project is to facilitate the growth of bacteria on various selective media to isolate all types of bacterial species. For my methods, I extracted the gut bacteria from my control and experimental group and streaked the bacteria on the respective plate media. To calculate the percentage of biodegradation, I inoculated a petri dish with a specific volume of bacterial solution at a fixed concentration. This was to ensure that the percentage of biodegradation was due to the type of bacteria instead of the quantity. These results show the average percentage of biodegradation between plastic-fed and non-plastic-fed waxworms. We can clearly see that plastic-fed waxworms had a higher average percentage of biodegradation at 0.793%. These results show the percentage of biodegradation among all my selective media as indicated on the x-axis. EMB had the highest average percentage of biodegradation at 1.384%. These are my qualitative results. I found many similarities between the cultured colonies of my bacterial species and Ideonella sacchensis. Another observation that really piqued my interest was the discoloration of plastic inoculated on the EMB media. I proposed that the bacteria took up the eosine methylene blue dye complex and precipitated it onto the plastic, which is why we see the blue smears. The percentage of biodegradation was not statistically significant between my media types or waxworm diet. But that being said, I clearly observed higher percentages of biodegradation on a specific bacterial species isolated on PEA agar, bacterial species isolated on EMB agar, along with those that were extracted from plastic-fed waxworms. In the future, I will conduct more trials to determine if this difference was due to random chance or the selective media types. To continue this research, I will identify the characteristics of the bacteria, observe the physical and chemical changes of the plastic, and possibly find an enzyme or bacteria that can be used in the process of bioremediation. My name is Rishika Podarala, and this is the isolation and identification of effective polyethylene degrading bacterial species from Galleria mellonella. Thank you.